Hello, everyone, and thank you for being at this talk. We would like to thank SPIE Medical Imaging for providing the opportunity to present our work. In this presentation, the topic is an observer study-based evaluation of a stochastic and physics-based method to generate oncological PET images. Positron emission tomography is a widely used imaging modality with multiple clinical applications, in particular for diagnosis and assessment of treatment response of cancers. Thus, several new and improved methods have been developed for PET image reconstruction, segmentation, and quantification. Objective evaluation of these methods typically requires the knowledge of the corresponding ground truth. For instance, evaluation of PET segmentation methods requires the knowledge of the ground truth tumor boundaries. Simulation studies provide a mechanism to objectively evaluate PET imaging methods, since the ground truth is known in these studies. In addition, simulation studies provide several advantages, including the ability to model in vivo anatomical and physiological properties, to model imaging system characteristics, incorporate patient population variability, and generate multiple scan realizations of the same patient to evaluate repeatability. Most importantly, all these can be done in silico, which is inexpensive and provide the opportunity to optimize the imaging methods before conducting clinical studies. Multiple studies have been developed to generate simulated PET images. However, for these studies to assist with clinically relevant evaluations, there is an important need to quantitatively evaluate if these methods yield clinically realistic images. For this purpose, one approach is to quantitatively evaluate the similarity between the distributions of simulated and real patient images. Evaluating this similarity in distributions is very important when using simulation-based strategies to train deep learning-based methods. For example, for the task of segmenting the primary tumor from oncological PET images, Liang et al. Developed a framework where the network was first pre-trained on simulated images with known ground truth tumor boundaries, and then fine-tuned on a small clinical training set. In this case, for simulations to effectively assist in training, it is important for the distribution of the simulated images to match that of the real patient images. This importance is also true when evaluating PET imaging methods using objective assessment of image quality-based studies. One key element in these studies is the description of the distribution of the objects to be imaged. And for the results of OIC-based evaluation studies to be clinically relevant, it is important for the object model to accurately capture the variabilities in patient population. Observer-based studies have been applied to evaluate the clinical realism of simulated images. Thus, we try to answer the question that if these observer-based studies could provide a mechanism to quantitatively evaluate the similarity in distributions between the simulated and real patient images. To answer this question, let us start with the evaluation of clinical realism using ideal observer studies. Let us consider an ideal observer that makes optimal use of all available information to perform the task of discriminating the real images from the simulated images. Let us denote the simulated PET images by f hat and denote the real PET images by f hat prime. In this observer study, we consider two hypotheses, namely H1 and H2, where H1 refers to the class of the simulated images, and H2 refers to the class of the real images. Thus, the simulated images are sampled from the probability of f hat given H1, and similarly, the real images are sampled from the probability of f hat given H2. For this ideal observer, we can define the test statistic as the ratio of the probability of f hat given H2 to the probability of f hat given H1. This ratio is also known as the likelihood ratio. The ideal observer then makes the decision by comparing the likelihood ratio to a threshold. For this ideal observer, as in Barry et al., using the statistical properties of the likelihood ratio and using the concept of a likelihood generating function, we can derive the relationship between the AUC and the probability distributions of simulated and real images. Essentially, this likelihood generating function, defined as G, provides a lower bound for the AUC, where G0 is the likelihood generating function evaluated at its origin. G0 can further be expressed in terms of the probability of f hat given h1 and probability of f hat given h2. From the first equation, we see that a lower bound of AUC equal to 0.5 is achieved by setting G0 equal to 0. And from the second equation, G0 is equal to 0 when the probability of f hat given h1 is equivalent to the probability of f hat given h2. Therefore, an ideal observer that yields an AUC of 0.5 leads to the inference that the distribution of simulated and real images exactly match. Implementation of the ideal observer would require the complete description of the probability distributions under both hypotheses. However, these are typically unavailable, especially when the task is to distinguish between the real and simulated images. Thus, we consider a practical but rigorous alternative, namely evaluation with trained human observer studies. Our goal here is to have an observer that sets an upper limit to the performance of any available human observer. And for this purpose, we specifically focus on a two alternative forced choice test, or 2AFC test-based human observer study. In these 2FC tests, a trained human observer is presented a pair of simulated and real images side by side at a time. The observer is then asked to select the image that they think is the real PET image. The observer implicitly computes two test statistics, namely theta f hat and theta f hat prime, and assigns the image that has the higher test statistic to H2. The assignment is correct if theta f hat prime is larger than theta f hat. So we can compute the probability of a correct assignment as the equation given here. As shown in Barry and Myers, this equation is the same as the equation to compute the AUC of the observer. Now, let us be reminded that in the ideal observer studies that we described earlier, an ideal observer that yields an AUC of 0.5 leads to the inference that the distribution of simulated images exactly matches that of the real images. Thus, in this 2FC test-based observer study, if a trained human observer obtains an AUC of 0.5, we may then infer that the distribution of the simulated images closely matches that of the real images. We now consider to apply this 2FC test-based observer study to evaluate a PET simulation strategy we developed in this study. This simulation study was conducted in the context of simulating the primary tumor in FDG PET images of patients with lung cancer. Liang et al. Previously proposed a physics-based projection domain lesion insertion technique that generates simulated 2D oncological PET images. This technique captures the observed variabilities in tumor properties from an actual patient population. Specifically, the tumor shape, size, and tumor to background intensity ratios were extracted from clinical images. In addition, the technique accounts for the variabilities in patient population by selecting the real patient images as the tumor background. However, this technique had limited ability to incorporate the variability in intratumor heterogeneity. This heterogeneity is often observed in tumors, and methods to quantify this heterogeneity from PET images and evaluate its clinical predictive and prognostic value is a topic of intense research. Thus, in this study, we advanced on this simulation strategy to model intratumor heterogeneity more realistically using a stochastic lumpy object model. We use the observation that the tracer uptake patterns within the tumor can be modeled as a combination of lumps, where the lump locations, amplitudes, and sizes are random variables. More generally, it is suitable to characterize the tracer uptake within the tumor as a random process. Thus, the intratumor heterogeneity was modeled using a stochastic lumpy object model, which is given by the equation here. In this equation, SR denotes the support for the tumor, N denotes the total number of lumps, Cn, An, and sigma n denotes the center, magnitude, and width of the nth lump function respectively. To model the tracer uptake as a random process, Cn was uniformly distributed within the tumor support. An and sigma n were uniformly distributed within the predefined range, but were scaled based on the tumor to background intensity ratios that are extracted from the clinical images. In the figure here, we show examples of the lumpy object model with different choices of parameters. Here, we show the representative simulated images generated using the simulation strategy we just described. In this figure, the high-resolution simulated tumor models with the corresponding reconstructed PET images were shown. These images demonstrate that the method can generate a wide variety of clinically observed tumor types, including small tumors, tumors with multiple hotspots, and tumors with necrotic cores. We observed that these images are visually realistic. Thus, the next step is to evaluate the clinical realism of these images by performing the 2FC test to quantitatively evaluating whether the distribution of these simulated images matches that of the real patient images. To perform the 2FC test, we developed a web-based app as shown in this slide. As previously mentioned, during the 2FC test, the trained readers were shown two images side by side at a time, one a real patient image and the other a simulated image generated using the PET simulation strategy we just described. The readers were instructed that the task was to identify the real PET image that they thought had the real tumor. Since this is a tumor discrimination task and not a tumor detection task, we provided the users the location of tumors in each image. To facilitate a robust observer study, the app incorporates functionalities provided by clinical software, including the option to invert the image intensities and the option to adjust the image contrast. To ensure that the trained readers involved in this test were indeed those who would set up an upper limit on any human observer, we considered 5 nuclear medicine physicians and 1 nuclear medicine physicist. Each trained reader performs the test for 50 image pairs. We then computed the fraction of times that each reader correctly identified the real patient image. During the test, the readers were also required to provide confidence levels for their decisions. The definitions of the confidence levels are given in the table here, with a confidence level ranging from 1 to 5, corresponding to from no confidence in decision to 100% confidence. In this slide, we present the results of the 2FC test. The table here shows the fraction of times that each reader correctly identified the real PET images, along with the median confidence level. We can see that the percent accuracy for each trained reader is only approximately 50%, suggesting that the distribution of the simulated images closely matches that of the real images. This figure shows the distributions of the confidence levels for 5 nuclear medicine physicians, for 1 nuclear medicine physicist, and for combining all the readers. The upper and lower rows show the distributions when correctly and incorrectly identifying the image pairs. When considering all readers, we can see that among the 156 correct identifications, only about 38% cases were with confidence levels higher than 3. This result indicates that the readers were not very confident, even when they made correct identifications. Finally, here we present examples of simulated images that were incorrectly identified by at least 3 readers, but with high confidence. This trend was observed over a range of tumor types, further demonstrating the robustness of our simulation method to generate realistic simulated PET images. So to conclude with the key takeaway messages, in this study, we provided a theoretical premise for an observer study-based framework to quantitatively evaluate the clinical realism of simulated images. This framework can be naturally applied to evaluate other image simulation techniques and is generalizable for other image modalities. The theoretical premise shows that an observer-based study could evaluate whether the real and simulated images are from the same distribution. We then applied this observer study to quantitatively evaluate a stochastic and physics-based method to generate 2D oncological PET images. And the results of the observer study demonstrated that the method generated highly realistic simulated images. In addition, the method demonstrates the ability to generate wide varieties of clinically observed tumor types, including those with complex intratumor heterogeneity. And pending necessary permission, we will publish the web-based app for the 2FC test for wider usage by the image science community. The demonstrated clinical realism of our simulation method motivates the development of this method to generate 3D PET images. Further, extending the method to statistically fit the lumpy object model using patient data will model the intratumor heterogeneity even more realistically. Finally, I would like to thank our collaborators, funding agencies, and also colleagues in my lab. Thank you again for attending this talk, and please feel free to contact me on the Slack channel or via email for any questions regarding this work.